Good morning, everybody. I hope everybody is doing fine, staying at home and following the instructions of the government for our safety. Now, as we all know that this lockdown may continue for 2 more weeks. So in order to finish our syllabus, we are going to classes. Any queries are there, please mention. We will try to solve these queries. Now, we'll try to rewind the things. As we all know that we are doing the study of recombinant DNA technology, RDT, or genetic engineering. So we have finished certain aspects or basic techniques of recombinant DNA technology, or the basics of recombinant DNA technology. What is this recombinant DNA technology? How it is useful for the mankind? And in order to study this recombinant DNA technology, we studied the basic aspect of recombinant DNA technology, that is tools used in RDT, where we learned about different enzymes like ligases, alkaline phosphatases, then endonuclease, and how we are going to use this restriction endonucleases for the purpose of genetic engineering. So we have studied about the types of restriction endonuclease. Remember, we studied two different types of vectors. One is plasmid vector. In plasmid vector, we learnt about the plasmid PBR322 and the plasmid PUC18 or PUC19 vector. We learnt about the advantages, disadvantages of these vectors, what are the properties of these vectors, and so on. Now, these are not the only vectors. Later, we have studied the bacteriophages as a vector. In bacteriophages, we studied about lambda bacteriophage vector. What are the different strategies? What are the different properties of this vector? How to use it? How to screen the recombinant vectors, and so on. Later, we studied something about M13 phage as a vector. So M13 phage, generally it is used when we need the single stranded copy of double stranded gene of interest. And in that scenario, we are going to use M13 as a vector. So we have seen different strategies to develop this M13 vector and the advantages, disadvantages, and so on. Now, next aspect of our vector study is when we need plant cells, or when plant cells are the host, what type of vectors can be used for plant cells. So in that regard, today we are going to study the structural organization of one plasmid called TI plasmid. Now, what is this TI plasmid? So this is one type of plasmid. It is a conjugative plasmid which is present in one organism called Agrobacterium tumefaciens. This Agrobacterium tumefaciens is a gram-negative organism which causes a disease in plant. So the disease which is caused by this Agrobacterium is called as crown gall disease, which is the uncontrolled division in the plant cell. So when this organism, gram-negative organism, that is Agrobacterium tumefaciens, infect plant cell, it causes some tumor kind of disease to the plants, and which is called as crown gall disease. And this disease, it is understood that this disease is caused by, or the property of this disease is given to this agrobacterium by presence of one plasmid called as TI plasmid, and hence the name is given tumor-inducing plasmid. The plasmid which is responsible for induction of tumor disease in the plant cell by agrobacterium. Now, when this agrobacterium is cured, or when this plasmid is removed from the agrobacterium, such cured agrobacterium cannot cause any disease to the plants when they are infected. Hence, the disease-causing ability, disease-causing property to this organism is given because of this plasmids called as TI plasmid. Now, there are different species of agrobacterium which causes different types of disease in plants. Another species is Agrobacterium rhizogenes, which causes hairy root induction or hairy root disease in causes, or which are responsible for causing the diseases in the plants by this Agrobacterium. Now, when we are talking about this TI plasmid, so TI plasmid is a megaplasmid. It's the size of this plasmid is huge. almost it is 200 kilobases plasmid. So it is called as megaplasmid. It is a conjugative plasmid. It means it can be transferred by conjugation. The genes, example tDNA, which will be expressed, or the regulatory sequences which will be recognized by only plant cells. The region on TI plasmid, there are certain regions on TI plasmid which are recognized by only plant cells. Means the regulatory sequences are there for the plant cell. It means these genes are expressed only in plant cells. Other than this, there are certain other genes which are having regulatory sequences in agrobacterium, and hence these genes will be expressed in agrobacterium, not in plants. So this TI plasmid will be, we can say we can divide the genes into two parts. One, the tDNA, which will be expressed only in the plant cell and not in the agrobacterium, and the rest of the genes or rest of the sequences are expressed only in the agrobacterium or in the bacterial cell. And hence this particular property is very important in case TI plasmid will be transferred to the plant cell, and it gives the oncogene property or tumor induction property to the plant cell. Now, when we are coming to the structure of this TI plasmid, depending upon the opines. Now, what are these opines? These are nothing but amino acid derivatives which are synthesized by the tDNA. So, depending upon type of opines, this TI plasmid will be divided into two types. One is agropine type and nopeline, octopine type and nopeline type. Now, if we see the structure of this plasmid, I hope you are able to see the structure. So this is a structure of this plasmid. It is a circular plasmid. It is a huge megaplasmid. This plasmid contains different regions. So what are the different regions? One is the tDNA. Which is responsible for transfer of oncogene property to the plant cell. So tDNA. Then there are certain other regions. Now what are the other regions? There are certain opine catabolism regions are there. That those regions are OC, that is octopine catabolism, agropine catabolism. These are the catabolism genes. So these genes will be expressed in agrobacterium, while the tDNA part will be expressed in only the plant cell. Now there is one region called TRA. Now what is this TRA region? It is the genes which are responsible for the conjugative transfer of this plasmid from one agrobacterium to another agrobacterium. Now, just listen carefully. The TRA region is responsible for transfer of this plasmid from one bacteria to another bacteria. Now, I am not talking about bacteria to plant cell transformation. It is bacteria to bacteria transformation. So, conjugation, through the process of conjugation, this plasmid can be transferred from one organism to another organism. And this happens because of the region, because of the genes called TRA genes. So TRA genes are responsible for conjugative transfer property of this plasmid. There is another region which is called as origin or origin of replication or ORI region. So this ORI region or origin of replication is for the agrobacterium. So this plasmid can be replicated in agrobacterium with the help of this origin of replication. Now there is one more region called as Vir region or Vir gene. Now what is the property of this Vir gene? It is also called as virulence gene. This virulence gene is responsible for the transfer of this tDNA part, only this tDNA part. The proteins which are synthesized from this virulence region are responsible for transfer of this tDNA to the plant cell. I hope you are understanding. So this TI plasmid is having different regions plant cell and causes crown gall disease in the plant cell. Now for this transfer, there are different proteins required or different regions which are required. One is Vir gene. Now, what is Vir gene? Virulence region or virulence gene, the proteins which are expressed from this virulence gene is responsible for transfer of this tDNA from this Ti plasmid to the plant cell. Now, other genes which are present on this Ti plasmid which are required for the agrobacterium organism. What are those genes? Say, for example, Tra gene. That is the gene which is responsible for conjugative transfer of this plasmid from one bacteria to another bacteria. When it comes to the conjugative transfer, this particular protein is responsible for conjugative transfer of this particular plasmid. Then, when this agrobacterium is growing in the plant cell, when this agrobacterium is present inside the plant cell, what it needs for the growth, for the metabolism, it needs nutrients. Now, how it will get the nutrients? Because the nutrients which are available in the plant cell may not be useful for the agrobacterium. And hence, for this reason, to get the nutrients, it is transferring its tDNA to the plant cell. Now, what this tDNA contains? The properties of tDNA, there are two different properties or two different genes which are present in tDNA. What are those two regions? One is the oncogene property. What is meant by oncogene property? Continuous cell division. When this oncogene property is given to the plant cell, so such plant cells will, without any restriction, such plant cells will continuously divide. So division property or oncogene property will be given to the plant cell by this tDNA. What is the another property which is given by this tDNA? There is something called opine synthesizing genes. So this tDNA contains two regions, two genes. One is oncogene and second is opine synthesizing gene. Now, what is meant by opines? These are nothing but amino acid derivatives. These are nothing but amino acid derivatives. Now, how this amino acid derivatives are useful? So, when this tDNA is transferred to the plant cell, when this tDNA is integrated into the plant cell will continuously divide because of the oncogene property which is given by this tDNA, and also it will start synthesizing certain amino acid derivatives. Now, which are those amino acid derivatives? The genes which are present in this tDNA. Now, octopine and nopaline, these are the different types of amino acid derivatives, opines, which will be synthesized because of transfer of this tDNA to the plant cell. So, when this tDNA gets inside or integrated into the plant cell, two properties are given to the plant cell by this Ti plasmid. What are those two properties? One, continuous synthesis of the cell or continuous cell division because of the oncogene property which is given by the Ti plasmid, tDNA of the Ti plasmid. And second thing, because of presence of this opine synthesizing genes which are the acid derivatives. So when these amino acid derivatives are getting synthesized in the plant cell, these amino acid derivatives are utilized by agrobacterium and it will grow and increase in number in the plant cell. So it will live happily in the plant cell because of this opines or amino acid derivatives which are synthesized by the plant cell. Now, to utilize this amino acid, there should be some genes for metabolizing or for catabolism of this amino acid to get the energy. So for that purpose, certain opine catabolism genes are present in the rest of the part of this Ti plasmid. So along with this Vir gene, along with the Tra gene, and along with this tDNA, what else is present in this Ti plasmid? It is opine catabolism genes. It means tDNA contains opine synthesizing genes and the rest of the plasmid contains opine catabolism genes. So when this tDNA is transferred to the plant cell will synthesize opines, while the agrobacterium which is present in that plant cell or which is present in that plant in that area will utilize those opines by using this opine catabolizing genes. So opine catabolizing genes will produce certain enzymes which are required for synthesis of this opines, amino acids, which are synthesized in the plant cell, and hence organism will grow. So it means the agrobacterium, when it infects the plant cell, with the help of virgin, the proteins which are synthesized by the virgin, it will cut this tDNA part, or it will transfer this, will start synthesizing certain opines, because there is presence of opine synthesizing gene in this tDNA. And because of oncogene property, the plants will continuously divide without any restriction, forming a crown gall. Now, because these opines are continuously produced, organism will utilize those opines, amino acids, by using this opine catabolizing genes, which are present in rest of the plasmid, and organism will grow in that plant cell. All right. So now this Ti plasmid is, we can divide this plasmid into two areas. What are those two areas? The region which is expressed only in the plant cell, and the region which is expressed in the bacterial cell. Now, which is the region which is expressed in the plant cell? tDNA. So tDNA doesn't have any sequences which can be recognized by agrobacterium. It means it will be expressed only in plant cell. In agrobacterium, this tDNA will not be expressed. But the rest of the region, that is opine catabolizing genes, vir region, tra region, and origin, these are the region which will be recognized by the agrobacterium, and hence these regions will be expressed in agrobacterium. So this Ti plasmid contains two areas or two regions which will be expressed in different cells. tDNA will be expressed in plant cell, while the rest of the region will be expressed in agrobacterium. And hence, because of this property, this agrobacterium is called as natural genetic engineer. Why it is called as natural genetic engineer? Because this agrobacterium is transferring certain genes from its Ti plasmid. So which are those genes? tDNA. So agrobacterium is doing what it is doing. It is transferring this tDNA, which is present in Ti plasmid within the agrobacterium, to the plant cell without any external help. It means naturally, with the help of this virgin, it is transferring this tDNA to the plant genome. So tDNA is getting integrated from this Ti plasmid, which is present in the agrobacterium, to the plant host genome. And hence, this agrobacterium is called as natural genetic engineer. Now, what are the problems? Can we use this naturally, whatever Ti plasmid is present in agrobacterium, can we use this for the transformation? Can we use it in RDT purpose? No, we cannot use, because there are three different problems associated with this. What are those problems? First thing, this plasmid, as I told you in the beginning, this plasmid is called as megaplasmid. Now, what is meant by megaplasmid? The size of the plasmid is huge. So it is very, very difficult to handle this plasmid. First problem. Second problem, it contains oncogenes. Now, I don't think anybody will wish that whatever plants they are going to transfer or transform, the plant will be they want that plant will be infected with the certain diseased condition. So oncogene property, which gives the disease causing property to the plants, I don't think that is required when we are using this Ti plasmid in RDT as a vector. So first problem is the size of the plasmid. The size of the plasmid is huge, and hence it is very difficult, which makes it very difficult to handle. Second, the oncogene property, which is the disease causing property, and when we are using this plasmid in the plant cell, we don't want this property to be there in the plasmid. And the third thing, this particular plasmid doesn't have the unique restriction endonucleosides. So these are the three problems associated with using this natural, as it is, Ti plasmid as a vector. So what are these three properties? Three problems. One is the size of the plasmid. It is a megaplasmid, so it is very difficult to handle. Second, it is having this oncogene property, which is a disease causing property, and we don't want this disease causing property to be there when we are using this plasmid for the RDT in the RDT as a vector. And third, because this plasmid doesn't have any unique restriction endonuclease site, it is difficult to cut the plasmid with the help of restriction endonuclease enzymes. So to overcome this problem, there are different strategies used. One is called as co-integrate vector strategy, and another is called as binary vector strategy. And those two strategies we'll discuss in our next class. I hope everybody is understood the organization of this TI plasmid, how this TI plasmid is going to help in RDT, how the mechanism happens in this Agrobacterium tumefaciens. And if there are any queries, please mention those queries in the box given below, so that I will try to solve these queries. Thank you all.